Dr. Michelle Dawson is an assistant professor at Georgia Tech in the School of Chemical and Biomolecular Engineering. Her research is focused on manipulating cell mechanics to control cell function. Quantitative microscopy techniques based on transport phenomena are used to characterize the economic signatures of cells, is that right? Which are important for cell migration, differentiation, and development in normal tissues and tumors. Genomic analyses and small molecule screening are then used to elucidate the molecular pathways controlling these mechanical profiles and to identify tissue targets for therapeutic development. Information gained through these fundamental studies can be used to improve the homing of stem cells to tissues, classify cells based on their mechanical properties, and increase our understanding of dose-dependent cancer progression. Additionally, they can use to optimize physical properties of engineered tissue. Manipulating the biophysical properties of cells can also improve the therapeutic response. This approach has been used to control stem cell differentiation and to make cancer cells more responsive to chemotherapeutic drugs. So thank you and welcome.
First, I want to thank you for giving me the opportunity to visit CSU. I'm really excited to be part of your faculty search and to have an opportunity to tell you more about my research. I hope that you'll find that it fits well in mechanical engineering. There's definitely a lot of mechanics in what I do, and that we're trying to understand how cells respond on a mechanical level. This includes force measurements, looking at stress on the surface, deformations. And so these are techniques that are similar to what you would do with a mechanical system, but with a lot of biological complexity that's added to understanding these types of behaviors. And so a lot of my work involves microscopy and using microscopy to image different elements of this cell, and then trying to quantify parameters based on microscopy and images. And a lot of my work is specifically related to cancer. All of us know that cancer is one of the leading causes of death in the United States. It's only second to heart disease, and it's not decreasing in the rate of mortality. And this disease is caused by multiple things, including mutations in several genes. These mutations then lead to the development of certain hallmarks. These hallmarks lead to mechanical changes in the environment and in the cells themselves. And those are the kinds of things we are interested in measuring. And I did not bring a pointer, so hopefully I can just use this. And so here you can see these are hallmarks that we associate with the development of cancer. Rapid growth, evasion of apoptosis, growth that's independent of normal environment constraints, and increased migration. We also know that tumors become more heterogeneous as they become more invasive. And this heterogeneous nature is associated with changes in cells, especially at the basement membrane, where the basement membrane breaks down and cells are able to escape and undergo metastasis. And that's what's shown here. And you can see the structure of these cells is very different than the more cuboidal cells that are within the tumor environment. And so we look at this kind of structural change as a signal that these cells might be more likely to undergo metastasis. We know that it's less than 1% of the cells within a tumor that are able to undergo this function. And so if we can identify these cells and cells that are more likely to have this ability, then we can hopefully look at different types of signaling processes that control it to come up with new drug targets that can be used to treat this disease. Because it's not the primary tumor that kills patients, but the metastatic tumors that leads to patient death. And mechanobiology is this study of how cells respond to the environment around them. This is through the interaction of proteins on the cell surface that interact directly with extracellular matrix proteins, either through things like integrins or through nonspecific binding. And what we do in terms of mechanical characterization is we look at forces associated with those mechanical connections or those physical interactions. Forces are really important in the development of cancer, and that has been shown in many studies. This is an illustration of a mammary duct, and you can see that a normal mammary duct is very symmetrical. It has multiple cell layers here, and these cell layers are arranged very uniformly, and then there's an extracellular matrix that surrounds the duct, and then there are cells that are like fibroblasts that are distributed within that matrix, and they secrete matrix proteins. But when a tissue undergoes tumorigenesis, or when a tumor begins to form in that tissue, it exerts stress, and that stress leads to changes in the tissue. And you can see that here as these arrows that represent different stresses on the tissue level. First, there's stress from the growth of these cells within the tumor, which lead to solid stress on the surrounding environment. And then there's the recruitment of host-derived cells that secrete these matrix proteins. And there's cross-linking of those matrix proteins, which leads to high stiffness within the tissue. And as things become more concentrated within the tumor microenvironment, so we have a lot of stress from the compaction of these things, then we also get compression of vessels, like blood vessels and lymphatics. And that leads to high interstitial fluid pressure. And so we have a situation where forces are accumulating. And these accumulating forces lead to signal activation within cancer cells. And that is one of the triggers for metastasis. And so understanding how these cells respond to these forces can help us to understand why cells undergo this pathological migration. On a cellular level, there are specific signaling pathways that are associated with these responses. One of the most well-known pathways is the Row-Rock signaling pathway. This is a pathway that controls actin, myosin contractility. And what we know is that when the tissue undergoes this stiffening response, which I've just described in terms of this cross-linking of this network and all of these cells that are accumulating there, that this stiffness is transmitted to a cell through proteins at their surface called focal adhesions. These focal adhesions are basically force transducers. And when the extracellular matrix stiffens, it transmits force through these force transducers, which activates this intracellular signaling pathway, leading to the expression of these intracellular proteins. And these proteins activate something called myosin light chain, which is then phosphorylated, and that leads to contraction of the intracellular filaments. And so these signaling processes are not acting alone, and so it's not just a force progression, but they're also connected to signaling pathways that control things like cell proliferation. And so when tumor cells undergo this mechanical change, it leads to increased proliferation, which further leads to increased force generation, and so we have a process that's like a runaway truck, where all of these things are accumulating and they continue to accumulate. And so that is one reason that mechanics is important in looking at how tumors develop. Another is that when we think about metastasis, we have a cell that's in the primary tumor that only forms metastatic tumors in certain tissues. And those tissues may be very far from the primary tumor, but it has a way of reaching those tissues and then colonizing those tissues and forming a tumor. And so to undergo this process, it has to deform to escape the primary tumor. It has to deform to move through the tissue environment. It has to deform to move through the vasculature. And then again, it has to deform to move into the secondary site and to form metastasis. So there are numerous deformations that are important in the migration of a cell from the primary to the secondary site and in the formation of a metastatic tumor. And we know that cell deformations are very similar to deformations of solid materials. And so we can look at this using stress-strain relationships. And when we think about a cell, we have to think about what is the intracellular component that is contributing to the generation of these forces. And a lot of that is caused by the polymerization or depolymerization of actin filaments, which give the cell its structure. These are some fluorescent movies of, oh, I'm sorry, what I just did. Sorry about that, guys. I was touching a screen and, okay. And so these are movies of
And
cells.
that is a smart board, so if you touch it, it will do weird things too.
Okay,
So be careful.
So these are movies of cells that are moving after they've been genetically engineered to express fluorescent proteins. In this case, this is GFP actin, which polymerizes to form actin filaments. In this case, these are 83 RFP, and so these are end-binding proteins that polymerize to form microtubules. And then the last one, we have genetically engineered cells that express GFP paxillin. And so we can see cytoskeletal structures that are dynamically changing in different cell populations. And so these are tools that we can use to understand when there's a change in force, what types of parts of the cell are involved in changing that force. And so we usually combine these types of tools with our force measurements to get a better understanding of what is happening on a subcellular level. We also know that these are cancer cells that are undergoing these changes, but on the right, these are stem cells that are recruited to the tumor microenvironment. And these are in control conditions, and these are after treatment with soluble factors that are present in tumors. And you can see that the organization of these cytoskeletal filaments, where here red represents actin, green represents microtubules, and blue the nucleus, you can see that the organization of these structures changes dynamically as well. And so it's not just the cancer cells that respond to the environment, but it's also the stromal cells that respond to the environment by changing things like their morphology, and their morphology is also controlled by mechanics. And so some of the tools we use, or some of the tools that we have in our toolbox, are things like traction force microscopy, which allows us to measure surface traction forces. This is a stress strain measurement where we're measuring displacements of particles that are embedded in an elastic substrate, and then we're using those to determine the stress profile. And that stress profile changes as the cell undergoes migration. And so this is kind of a low resolution video of the cells moving and the associated forces. These are images of cells moving on a surface, and we can do things like track their nuclei so that we can understand not only the force profile, but what the motility of the cells actually looks like as these cells are expressing forces that are of interest to us. We can also embed particles within the cell fluid itself or within the nucleus, and then we can use those particles as probes to look at the mechanical environment within the cell. And so we do a lot of these things simultaneously to try to get a better understanding of how the mechanical properties within the cell correlate with the surface traction forces that the cells exert on their surface or on their exterior. And this is an image of cells that are labeled with a dye that is reduced in terms of its intensity as the cells undergo division. And so we can also use probes at different functional parameters so that we can determine whether or not there are things that we can add to our understanding. So not only are we looking at mechanics, but we're looking at cell proliferation, which is really critical in cancer cells that are undergoing more rapid division than normal cells. So this is another slide that goes over traction force microscopy. The red dots here are polystyrene particles that are embedded in a polyacrylamide substrate. Polyacrylamide substrates are often used because they're perfectly elastic, so it's really easy to determine traction forces for cells that are moving on these perfectly elastic substrates. And when we have the cell on the surface of this gel, it exerts forces, but when we detach the cell, those forces are released. And so we can take a stressed image and an unstressed image and use that to determine the motion of the particles from that stress within the substrate, and then use that to determine a strain field. And then that strain field can be related to the stress using Cree's function. And so this process is something that we do using MATLAB, and so we have generated code that allows us to analyze this for hundreds of cells or for populations that are of interest to us. And what we end up with are heat maps of the traction stresses. And this is a really interesting heat map because it shows in blue the regions of lowest stress and in red the regions of highest stress. And you can see that high stress is isolated to one side of the cell because the cell is polarizing these forces, which happens as the cell is undergoing migration. So not only do we measure the stress that is exerted on the surface, but we also measure whether or not those stresses are being polarized or the forces are being polarized, which is essential for the cells to actually undergo translation. And so to look at polarization, we just look at the center of mass of the cell itself and then compare that to the center of mass that's weighted by these forces. And that gives us an idea of whether or not they're truly polarized. The other technique that we use often in the lab is particle tracking methodology. And I already mentioned this a little bit. And basically we use a ballistic particle injection system to inject particles into the cytosol. This bypasses endosomes so that the particles are embedded within the cytosolic fluid and not taken up by cytoskeletal filaments. When particles are moving along filaments, they move by directed motility. And we don't want that. We want the particles to be moving randomly within the cell so that we can use them as probes of the fluid properties. And
And so in this case, these are murine MSCs that have been injected with 100 nanometer particles. Then we use high optical resolution to image those particles, and then we track the motion of the particles based on the motion of their centroid. And from that, we can get traces of the motion, and we can relate that to fluid properties based on the slope and the amplitude of the mean square displacement, which is the change in the location of the particle with respect to time squared. And so for a particle that's moving by random motion, where you get these kind of large displacements, you see mean square displacements that vary linearly with time, indicating that the particle is moving in more of a viscous fluid. Whereas when a particle is trapped, its motion is restricted, and this restricted motion gives us a mean square displacement that doesn't increase with time because the particle's not moving. And so then we can associate that with more elastic fluid behavior. And so this is the kind of measurement we're making, and then we can make a lot of downstream analysis based on these types of measurements. And so in terms of tracking cells, this is a really powerful tool that we use all the time. One thing that's great about it is we can simultaneously track hundreds of cells for 8 to 10 conditions, and then use that to determine how the cells move in control conditions, and then how they move after some chemical is introduced, or after they've undergone some sort of physical change. And so this gives us an idea of whether or not certain things lead to increased motility in cells. And these are some data for SCA3 cells, which is a highly metastatic ovarian cancer cell line, and OVCAR3 cells, which is a non-invasive ovarian cancer cell line. And each one of these dots represents the migration coefficient for a single cell. And you can see that on average, the migration coefficient is higher for these SCA3 cells on the soft surface. But there's this population of cells that moves much faster than even the average. And so the beauty in this technique is that not only do we measure an increase in the average migration coefficient, but we can look at subsets of particles that might be more likely to undergo metastasis. Another thing that's important in the context of cancer is whether these cells undergo what's called an epithelial to mesenchymal transition. And so most of your cells in your body are epithelial on the surface, and that means that they form these nice structured tissues, and they kind of form cuboidal surfaces, and they are well aligned. But when a cell undergoes a change to a more mesenchymal phenotype, it leads to changes in the expression of certain cell adhesion molecules, which we can look for. And then it also leads to changes in their shape. Like they become more elongated, and they detach from surfaces. And so these changes are associated with cancer and with highly metastatic tumors. And so we've been really interested in how the epithelial to mesenchymal transition affects the forces that cells exert, and whether or not we can use that information to our advantage in understanding cancer or in finding new ways of treating it. This is an example of how we've used all of these tools together to look at differences in populations. And I want to talk about this before I go on to giving you some specific examples, because it nicely summarizes how we can fit this information together. And the cells that we used in this study were MCF7 cells, which is a non-invasive breast cancer cell line. And we have control cells, which are the non-invasive breast cancer cell line, and then we have cells that are genetically engineered to express SNAIL. SNAIL is a protein that's a transcription factor which is associated with EMT. And so these cells have undergone a mesenchymal transition, and so they have this more metastatic phenotype. And you can see the structural change in the cells. And then we use traction force microscopy to look at changes in force, and you can see a change in force expression. And then we use particle tracking microbiology to look at intracellular mechanics, and the cells actually become softer, which is associated with less polymerized actin, which we show here, and then the cells have increased motility. And so we have a cell now that has this more mesenchymal phenotype. It expresses higher forces, it has a softer intracellular behavior, and it has increased motility. And that's true for many invasive cancer cell lines. But this force profile may not be true for cells that are not invasive cancer cells. And so like for stem cells, it's very different. And so my talk today is broken into 3 subjects. The first is looking at how soluble factors within a tumor microenvironment affect cells within that environment. And these studies are primarily focused on looking at stromal cells and how stromal cells respond to soluble factors that are secreted by tumor cells. The second part of my talk is focused on matrix mechanics and how cells respond to higher stiffness. We know that increased stiffness is associated with cancer. And so looking at how cells respond to stiffness in these environments in this simple study allows us to see whether there are certain cells that are more likely to respond to these cues. And then the last part of my talk is focused on taking these two different types of cells and looking at them together and trying to understand whether or not some of these perturbations that we see in these simple models are also happening within these more complex environments. And so for the studies on stromal cells, we have used bone marrow-derived mesenchymal stem cells, which we isolate in the lab or we purchase from a regenerative medicine center, depending upon whether they are animal cells or whether they are human cells. They're isolated from bone marrow from normal donors in the case of humans, and from mice that don't have tumors, or rats. We have some rats, but all the data I'm going to show today is either from mice or human-derived bone marrow-derived MSCs. And the reason that we think that it's really important to look at this cell population is because these cells are recruited very early on to tumors. And they have a lot of implications in what happens in the development of the tumor microenvironment. We know that they interact not only with the cancer cells, but they interact with immune cells in the environment. And we know that those interactions are somewhat controlled by soluble factors. And a lot of downstream things that happen when these stem cells are present in these environments are higher incidence of EMT, reduced apoptosis, increased angiogenesis, and suppression of the immune system. And so not only are they recruited to the tumor, but they play these kind of pathological roles in the tumor. And so understanding how they respond to factors in the environment is very critical. And these are MSCs that are in control conditions, so just normal FBS-free media, and then MSCs treated with growth factors, also in FBS-free media. And this was the first study we did with these cells, and this was really groundbreaking for us because we took these cells and we treated them with tumor-conditioned media from FOLB-C derived tumor called 4T1, which is a mammary tumor model. And within 24 hours, you see this morphological change, which I already showed you. But this data is over an hour. And this is particle tracking data, which shows the changes in intracellular mechanics. And I already told you about how if a particle has motion that's associated with linear mean square displacements, that means that it's moving in a more viscous fluid. Whereas if the particle motion is inhibited, it's more elastic. And so you see within 60 minutes, the fluid within the cell is completely transitioning from being viscous to being elastic. And it happens within this very short period of time, much faster than this morphological change. And so particle tracking allows us to see what's happening within the cell before we could ever see it by eye. And these are some of the things that happen downstream of this mechanical change, including elongation of the cells, changes in the expression of focal adhesion proteins. In this case, green is vinculin, which is one of the proteins that's present in a focal adhesion. Changes in cytoskeletal structure, again shown with actin and tubulin. Motility in 3D gels. And then we took these cells that had been treated with TCM and we injected it in mice that had tumors. And more of these cells were able to be recruited to the tumor. And so it actually increases the homing of the cells directly to the tumor. And so we think that these kind of mechanical changes that we see within a short period of time are really important in directing their motility in vivo. We wanted to find out whether this behavior was special, whether we would be able to replicate this behavior in another stromal cell. And so we looked at fibroblasts and we wanted to see if they respond similarly. And what we found is that fibroblasts do respond to TCM by undergoing a stiffening response, but it takes them 3 hours to undergo the stiffening response. And when we look at the morphology 24 hours later, they never undergo this morphological change. And the migration in comparison is much lower. And so we think that this early mechanical response is really critical in increasing the motility of these stem cells that are derived from bone marrow and in leading to these morphological changes that are so profound. We wanted to find out whether there were certain soluble factors that were present within the tumor environment that caused this. And so we looked at several soluble factors, and the study I want to tell you about is focused on 2. platelet-derived growth factor, which is really important in the vasculature. It's also important in tumor environments. It's one of the things that's released from degranulated platelets, and transforming growth factor beta 1, which is highly expressed in tumors and has been associated with more invasive cancers. And so we focused on these two because they are also both involved in fibrotic disease, and so we thought maybe that these might be good factors to look at in the context of this stiffening response. And so we did some studies looking at the morphology of the cells after treatment with these factors, and we see that the morphology changes in response to all of them. And sometimes that morphological change is pretty drastic, like we look at things like changes in cell and nuclear shape factor. This is a change in cell shape factor, and you see that when we treat with TGF-beta1 or the combination of TGF-beta1 and PTGF, we have high changes in this. And we also see more actin stress fibers. We then used microbiology to see if there were changes in intracellular mechanics. And what we found that was really interesting was that the cells under control conditions, of course, are viscous, like we saw before, but when we treat with PDGF, we see no difference. Then when we treat with TGF-beta1, we get the stiffening response, similar to what we saw with TCM, but if we add PDGF and TGF-beta1, then we get increased stiffening response. And so PDGF, although it doesn't cause stiffening on its own, enhances the response that we see to TGF-beta1. And so we were really interested in the crosstalk between these signaling pathways. And so we treated with inhibitors of either of these receptors for these two signaling pathways. And we found that if we inhibited either pathway, that it reversed this mechanical response. And so we looked to see what was happening when we treated with TGF-beta1. And we found out when we treat with TGF-beta1, PDGF is actually secreted. And it binds to the receptor. And so if we do a binding assay to see how much of the receptor is binding to an antibody, we have less binding on the antibody, indicating that it's already bound to the ligand that's being secreted. And so that was our confirmation that it was the activation of the signaling pathway from TGF-beta1. And so when we saw this immediate stiffening response, we were just accelerating that process. And so another thing that adds more interesting information to this story is that if we treat with PDGF, it increases migration. Either random migration, like I showed you before, when cells are moving on a surface, or migration when cells are moving in a directional way, like as if they were closing a wound tissue in a scratch assay, like here. And so you can see in PDGF-treated cells, they migrate more, but when we add TGF-beta1 to PDGF, when cells are moving directional, then TGF-beta1 enhances the PDGF-directed motility response. And so these factors have crosstalk that also plays a role in motility. And so now one of the things that we're really interested in doing is looking more spatially at how these forces change during this motility process. And so one of my goals is to be able to simultaneously measure the intracellular mechanics and the surface traction forces, as well as the motility, so we can see as the cells are undergoing these changes, whether the intracellular mechanical change comes first or whether the surface changes come. And so we can look at these things together with some of the tools. But one of the major barriers to this is increasing our resolution with our microscopy. And so one of the things that we need to do to really make this happen is probably to use something like a spinning disc focal microscope. We may be able to use super resolution microscopy to solve this problem as well, which would allow us to look at individual particles with higher spatial resolution as well. And this work is something that I'm planning to submit to NSF Physics of Living Systems. So that's the first future project that I have kind of inserted along the data path. And the next story I want to tell you about, which is also under the heading of soluble factors and how soluble factors affect stromal cells, is work that we did trying to replicate this in human cells. And when we started this project, we kind of expected that they would behave the same way as the mouse cells did. But what we found was that the intracellular mechanical properties, the mechanical response to soluble factors, was not uniform in human cells, but it was uniform in mouse cells. So when we track particles in a mouse cell and we treat with tumor-conditioned media, every particle senses a stiffer environment. But when we do the same study in human cells, only some of the particles sense a stiffer environment. And if we look at low passage cells, a larger percentage of those particles sense a stiffer environment. And if we look at high passage cells, that number is reduced. And so we see that with passage, there's reduced mechanical response in these cells. And so I asked my students to go back to the cells and find out whether it was overall changes in kind of the spatial information within a cell, or whether it was certain populations of cells that were not responding. And what he found was that it was certain populations of cells that were not responding, and that population of cells was basically just increased in high passage cells. And so we did some more clinical or preclinical studies to see what effects this has downstream. And in this case, we really focused these studies on wound healing, and to see whether or not these cells could close a wound. And so what we found is that the low passage cells contract wound tissue. And so for these studies, we take stem cells that are labeled with a red dye, and we inject them at the periphery of the wound, and then we wait for the wound to close. And with the low passage cells, the wound is contracted, but with the high passage cells, the wound is not contracted. We also saw that the migration was reduced for the high passage cells, and their ability to contract collagen gels was reduced. And so it was a loss of function that occurred when they lost this ability to respond to soluble factors by altering the intracellular mechanics. And so the mechanical measurement we were making within 1 hour was very useful in predicting the behavior of these cells in these much more complicated situations. So how are we going to use this information? And so one of the questions that I'm really interested in is, how does this in vitro aging compare to in vivo aging? We know that cell senescence occurs as cells undergo increased numbers of passages. Well, cell senescence also happens in vivo. And so we want to find out whether or not these kinds of loss of mechanical changes that we see is something we also see in cells that have been isolated from older donors. This is really important in the context of cancer, since age is the #1 risk factor for the development of cancer. And we can see higher incidence of cancer in patients that are in these higher age ranges. And so for this project, I have a few questions that I would like to answer. One is, how does aging affect MSC mechanics? And so for this, we can define the molecular and mechanical differences in MSCs isolated from premenopausal and postmenopausal donors. The second question is, how does this contribute to malignancy? And then we can expose them to tumor-mimicking factors, like tumor-secreted soluble factors, and see if they respond the way we saw before with the cells that we were culturing in vitro. And we can also look at changes in gene expression and changes in molecular signaling pathways using molecular inhibitors. And all of these things we've done in the context of other questions. And so these are things that we've been doing for a while, but now being applied to a new problem that is of interest in terms of human health. So the next set of studies I'd like to tell you about are related to how cancer cells respond to changes in the stiffness of their environment. And we've done a lot of work looking at breast and ovarian cancer, which are two types of cancers that specifically affect women. And a lot of my research is really focused on women's health, and that has just been an area that I've just kind of gravitated towards naturally. And one of the questions we have is, how do metastatic cancer cells with these very different behaviors respond to changes in the mechanics of their environment? And we focused on breast and ovarian cancer for an important reason, and that's because the metastatic process is very different in these two tumor models. Breast tumors metastasize